The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
This month, can you teach compassion? And when you try, what does it do to your brain structure? Also, why fewer women publish in the world's best science journals, and how quickly do populations evolve away from social traits to block the spread of disease? Hello, this is episode 89 of the eLife podcast. I'm Chris Smith from The Naked Scientist. And first to a biochemical detective story about an extinct species of giant sea cow called the Steller sea cow that inhabited the frigid waters of the Arctic until about the mid-1700s. How did an animal closely related to a warm water dweller like a manatee survive in these sub-zero climes? Part of the answer lies in a tiny switch in its blood haemoglobin structure, which enabled it to more efficiently release oxygen to tissues despite these very low temperatures. The discovery is the work of the University of Manitoba's Kevin Campbell.
The story of this creature actually goes back to 1741, when it was discovered by naturalist George Wilhelm Steller, who was actually the naturalist on the ship captained by Captain Bering, that you may have heard of, the Bering Strait and Bering Sea. They were shipwrecked on an island off the coast of Russia, a really cloud-shrouded, cold, desolate place, where they discovered Steller sea cows, which became known as Steller sea cows after Steller himself. These animals are very interesting in that they're very closely related to manatees and actually more closely related to the dugongs, but they were quite different in that they were immense. They were whale-sized. They could be up to 10 meters long and maybe up to 10 tons. They lived along the rocky, desolate, cold, wavy shores of these Commander Islands, and this is the last known population, but they had lived previously in the Alaskan archipelago. And there's not the same food that is usually eaten by manatees and sea cows, which is seagrasses. They actually became adapted to eating mostly kelp.
And where have they gone?
They were actually probably on their way out. So estimates are that when Steller discovered them, and again, this is the last remaining population of this species, that there was maybe 1,000 to 2,000 individuals at most at that point in the early 1740s. This species was probably, on its way out already, though sadly they were hunted to extinction only 27 years after their discovery, mostly by shippers. People would stop off the islands and get some very tasty Steller sea cow meat.
And so how is it that 250 years later you're able to study them?
The conditions on these Commander Islands, they're rather cool, and so some of the bones have remained washed up on shore and are now in museums. They're of good enough quality. We were able to start getting some DNA. And of course, we targeted hemoglobin genes. But now, even the last couple of years, there's been a complete genome of this species.
Why did you go for hemoglobin?
Well, hemoglobin is actually a really interesting molecule in the sense that it's the molecule that carries all the body's oxygen from the lungs to the tissues. So it's actually one of the few molecules that really links animals with their environment. Like most enzymes or most other chemical reactions, it kind of works a little bit better at offloading oxygen at warm temperatures, but not so good at cold temperatures. At really cold temperatures, it tends to hold on to oxygen really, really tightly. And this can be a problem for Arctic animals or Arctic mammals or birds specifically, because they often let their extremities really cool down to close to 0 degrees if possible, so that they can minimize heat loss. And by minimizing heat loss, it actually lowers their overall energy budget. So they actually have to eat less, right? So it's kind of like, imagine your house in winter. If you kept the windows open, you could keep your house warm, but your energy bill would be enormous. But if you can modify your hemoglobin so you can lower the temperature at which it's still able to offload oxygen efficiently or efficiently enough, they can save a lot of energy.
And you have, through the genome, got the genetic sequence, the recipe effectively for their hemoglobin, and does it show adaptations or differences?
We studied 3 different Steller sea cows, and we found a very unusual mutation in their hemoglobin, and we found a change in the beta subunit at a site that is perfectly conserved in all other mammals. At this one position, it's a lysine, which is a very strong positively charged amino acid. But in Steller sea cows, it got changed from a lysine to glutamine, which is a neutral charged molecule.
And what do you predict that would do to the haemoglobin in the Stellar sea cow?
Well, fortunately, there are some humans that carry the same mutation. It actually causes an increase in the overall blood oxygen affinity.
The problem that the Stellar sea cows are grappling with is in the cold, they want to give up oxygen from their bloodstream into their tissues, but without having to have a ferocious rate of blood flow to get enough oxygen in. So they want their haemoglobin to give it up readily. If it increases the affinity, this change of the haemoglobin for oxygen, does that mean the haemoglobin hangs on to it better? In which case, how does that serve their purposes?
That's the one single big difference between human hemoglobin that has this change and stellar sea cow hemoglobin that has this change, in that in humans, this effect causes the affinity to go up. But in stellar sea cows, this change causes their blood oxygen affinity to actually go down. And a second thing is, it takes the same amount of heat to break this energy bond between oxygen and the hemoglobin, regardless of the hemoglobin type, right? Whether it's from a reindeer, a stellar sea cow, a woolly mammoth, or a human. However, these cold adapted species tend to have some modifications in their hemoglobin. So, as I mentioned, it takes heat to break bonds, but hemoglobin doesn't only just bind oxygen, it's able to bind other molecules. And we have a organic phosphate in our red blood cells called diphosphoglycerate. And it's also able to bind to this molecule. But these molecules tend to bind to hemoglobin when it is ready to offload its oxygen in the tissues. And the interesting thing is when these molecules bind to hemoglobin, it actually releases heat. So in colder temperatures, these hemoglobins from these cold adapted species tend to have these modifications where these other molecules can bind to hemoglobin, and that releases some heat, and then that heat can actually get donated or kind of shifted over to the where the oxygen is found, and it's a little almost a self heater to help break that bond between oxygen and the hemoglobin in the cold tissues of these species. But the change in the stellar sea cow was really perplexing to us at first because it actually lowers the amount of diphosphoglycerate binding to the molecule, which is what we think usually gives the heat. But this change, as I mentioned, is found right in the central cavity of hemoglobin. And so what this change does itself is it lowers the amount of heat required for this change. And so this kind of gives that energy itself to break the oxygen bond. So it's the precise opposite as what we find in most other mammal species. They're doing a different mechanism, but the end goal is they're able to offload oxygen pretty good at cold temperatures.
Kevin Campbell. And one exciting spin off of that discovery, Kevin tells me, is that it might enable us to make blood substitutes that could keep tissues oxygenated at very low temperatures, such as during organ transplants. Let's hope so. In the year 2000, Eleanor Maguire published a paper that caused a seismic shift in the neuroscience landscape. She scanned London taxi drivers as they completed the knowledge, the arduous task of learning every road through London. She famously found that the brain region known as the hippocampus in these drivers enlarged as they progressed through their training, showing that even the adult brain has impressive powers of plasticity. So what about other aspects of behaviour, and in particular social skills and compassion? Can these be taught? And if so, what changes, if any, are visible in the brain in the aftermath? From the Max Planck Institute, Sophie Volk.
Can we train social skills to adults? Can we learn to be more compassionate, more understanding for others? And if so, what would this do to the brain? And we had different types of mental training, such as trainings that you know from maybe yoga practice, mindfulness, social training where you speak with your partner about your emotions and the other person listens and then the other person speaks about their emotions. Or also that you learn that you have different perspectives in yourself. So, for example, I'm a lab leader, but I'm also a mother, and I'm also somebody that loves to watch crappy shows on Netflix when I'm tired. So I have all these different perspectives in me, and as do you all. And learning about them helps you to understand the different roles we all have. And hence, it may increase the understanding we have for other people and their perspectives.
It's sort of practice making perfect then. So by practicing to be better at this, you in some way change the way your brain processes information, and its structure and how it handles information does change in response to those stimuli.
That's the idea, yeah. To let people increase awareness and also performance in emotional understanding and compassion, but also taking the perspective of others.
There's this old saying, isn't there? You can't teach an old dog new tricks. And I suppose that's really relevant with the brain. We do learn much better when we're younger than when we're older. So who did you look at, and how did you actually make these measurements?
We recruited adult individuals between 20 and 55 years of age, normal, healthy adults, willing to take part in this rather intense 9-month training protocol. So it ended up being like both men and women, average age 40, but distributed between 20 and 55.
And what did you do to establish the baseline? How did you work out how their brains were working before you did anything to them?
For the beginning of the study, we did many behavioral tests to kind of establish a baseline. And then we scanned them to understand the structure of their brain, so their anatomy based on gray matter and white matter, but also look at the function of the brain, namely looking at the activations that happen over a certain time period while you're scanned.
And then you subject your subjects to a fairly, as you put it, intense training over the 9-month period. Did you look during that period, or did you look just at the end to see whether anything had changed?
Because it was 3 different types of training, because part of the study, the goal was also to understand if there's differences in learning to understand other people's emotions, further understanding their perspectives of thoughts. So we wanted to also compare different types of trainings to understand the specificity. So everything was a 3-month course, and every 3 months of training block was then evaluated by having a set of scans, also a set of behavioral measurements, so that we kind of track how people changed over time.
And on average, we can look at how individuals might have performed differently in a minute, but on average, what happened over the course of the study?
Many things happened. I think focusing more on brain structure, you see over the course of 9 months that particularly the structure on average changes, but function is more subtle, and it seems to adapt more to the circumstances or the specifics of what you train. So here, what I observed in the functional measurements that I took, it wasn't something that just kept on adding, but something that kept on dynamically changing depending on what the needs of that certain training protocol were. So over 9 months, brain anatomy changes in a more slow fashion or a gradual fashion. It builds up upon each other, whereas function is something that you get a bit more of this if you train one thing, but then this reduces again if you train another thing.
We had an insight that this sort of thing happens because of the famous London taxi driver study with people developing larger subregions of their hippocampus if they have to do the knowledge and learn the streets of London to navigate them very well. So what does this add? Does this tell us that it's not just the memory and navigational circuits, it's the emotional and other circuits in the brain that change as well when we use them?
It depends on how you look at it, but for me, it means that we can still also change at adults, right? So the brain is like a sponge, and depending on the environment and the opportunities that we give ourselves, we can always learn and adapt to the circumstances, always within certain limits, and maybe over time with age and with certain habits or certain genetic predispositions, the opportunities to change are like more limited or different. So across individuals, but still the context matters to the brain, and the context is part of what shapes how the brain looks and functions.
Many would argue that's really encouraging to hear, because some of your subjects were older, so we don't seem to be completely fixed in our ways. I mean, maybe there were some subtle effects if you looked at just older people versus younger people, but what are the implications for people who have neurodevelopmental disorders? I'm thinking of people who might be on the spectrum, the autism spectrum, for example. If the brain can be, I don't want to use the word reprogrammed, but if it can be remolded in order to make it function better under certain circumstances, does this mean that we should be offering certain types of interventions to people with some of these particular developmental situations to help them?
Yes, the question is to what extent you can support that have neurodevelopmental conditions, because it was like obviously one should always stimulate somebody, right? Like if somebody is sick and starts feeling better, you should also encourage them to get out of bed, right? And get on moving after an operation. And probably this is the same, that you have to find a good way to stimulate people, but not all stimulation is right for whatever occasion or whatever person. And I think one of the take homes also of the study is like how differentiable it is, so that you can also train different things depending on what you need, such as more emotional skills or more understanding of perspective skills, but also this more mindfulness, attentional skills. And now often these things are sometimes combined together in different ways, and by sort of unfolding them and seeing how these components actually contribute differently to the brain, but also to behavior. I think it's also a first step to maybe understand like what components could then be best tested for a certain individual.
So there's hope for us all. Even in adulthood, we can change our brains. Sophie Volk there. This is the eLife podcast, and I'm Chris Smith. In a minute, how does the spread of infection alter how populations behave, and can biochemical clocks that predict disease risk in adults be applied meaningfully to children? But first, if you're a top scientist with an amazing discovery to tell everyone about, you send your manuscript to the world's best journals, don't you? Well, apparently not. If you're a woman, McGill University's Vincent Lariviere has found that female scientists are much less well represented in the top-tier publishing houses, favouring instead publication in worthy but less glitzy venues.
Many studies have shown that there's a gender gap in science, that there's less women than expected. And we also observe that women are less likely to receive, let's say, a lot of credit for the science that they do. So one of the markers of credit is citation. So is your research used in other papers that are being published afterwards? So what we try to do is to try to understand a bit more this gender gap in science by looking at women and men's publishing patterns. Where do they publish their findings?
And where do they publish their findings?
Well, unfortunately, they are globally submitting their findings in journals that are less prestigious. So in science, where you publish matters. And there's a premium in publishing in a certain set of journals that are very visible, very international, such as Science, Nature, and PNAS. And so what we try to do is looking at whether women were more or less likely to submit their findings to these very elite journals.
So it's not that the women are sending the work into the journal and it's being bounced. It's that they're not sending it there in the first place, potentially.
Exactly. So women and men's paper are actually accepted or rejected at the same rate. There's no differences in the proportion of women's paper that actually gets rejected. However, women are much less likely to send their work to these elite journals. And the reason for that, and this is actually quite depressing, is that they were considering their work to be of lower quality. So it's a self-perception of their work that makes them not submit their work to these journals.
We're slightly putting the cart before the horse because I haven't asked you yet how you arrived at these findings, but it's not just for clarity that the women are doing science that's of less interest to these journals and therefore they're not sending them there. It's that the work could be sent there. It's of a sufficient standard and interest, but they're just not choosing to send it to top-tier journals.
Exactly. And the main reason there is that they considered their work to be not as groundbreaking or sufficiently novel.
That's a pretty big claim that you're making. So how did you arrive at that conclusion?
So we basically surveyed about 5,000 authors of scholarly papers, both men and women. And we basically asked them, did you ever submit to those journals, which are Science, Nature, and PNAS? We also asked them whether their papers were accepted there. And we also asked them whether they were, in cases of rejection, whether they were desk rejected or rejected after careful, let's say, review from the journal. So it's really through a survey of, again, several thousand researchers. And again, the finding that we had about submission was observed in every field. So in medicine, in the natural sciences, and as well as in the social sciences, women being less likely to submit to these top journals.
And did you ask the women why they hadn't sent their work to these journals when it was worthy of being featured there.
Yeah, exactly. Women were much more likely to say that their work was not of a quality that was as high. They were more likely to say that they considered their work to be not as groundbreaking.
Do you think, though, that to a certain extent this represents caution? Men traditionally are risk takers, especially younger men, and women tend to be, on average, a bit more cautious. So is it that they're thinking, well, I could go for the top tier journal, I might get rejected, there's a high rejection rate there, and then I'll end up wasting time having to send it again to somewhere else, so I'll take the cautious, surefire approach that will get me published. Whereas the blokes will just say, no, I'll chuck it into the top tier journal, I might get lucky.
Absolutely. So that's one of the main hypotheses that we have that would explain the results. But then we need to think about how to mitigate to improving this gender gap that exists.
And what sort of strategies might there be? What, better mentorship, just encouragement, journals actually going out there on a PR exercise saying, hang on a minute, you should send your work here?
Yeah, exactly. So there's, on the one hand, such policies that aim at encouraging women to submit their best work to the best journals. But there's also another angle, which is to stop using the journal as an evaluation criteria. We now understand that there are biases in what these elite journals publish. And so by using indicators such as the journal impact factor, which is an indicator of the global citations and impact that a journal receives. So by using that, you're actually reinforcing the various disparities that exist in the system. So I would lean towards a decrease in the usage of journal level indicators to assess researchers and try to move to more holistic ways of evaluating scientists.
Vincent Lariviere there. For more than 2 years during the COVID pandemic, the majority of us had to fight our natural instincts to socialise and instead stay away from those that we love and like to mingle with normally to break the chain of disease transmission. Social behaviours evolve and become entrenched in populations because they can confer great benefits: support, defence, food, growth. But if a disease emerges in a group, how quickly can an evolved pro-social trait be shed to achieve nature's own equivalent of social distancing? Pratik Gupte.
What happens to a population of individuals that have evolved rules for how to be social in the absence of a pathogen? When an infectious disease then enters their population, how does that change their behavior, especially in terms of being social when a disease first enters a population?
I suppose it's a balancing act, isn't it? Where you've got the benefits of being social, where there's learning from other individuals, there's safety in numbers, there's exploiting food, versus if an infection gets into that group, there's a price to pay. And it's the balancing act of those two things.
Yeah, absolutely. So what we've done is to say, well, each one of you can choose how you balance the costs and benefits of being social. And so what we found there is that you get a split. Some individuals will say, I want to be completely safe and I'll avoid everybody else. So that's very much like what you would think of as shielding during COVID. But then there's other individuals that say there are serious benefits to being social, such as finding food. I will follow some of the other individuals that I can see, especially if they look like they've been successful in finding food themselves, and risk it. I will take the costs. And the only reason that these two can exist sort of side by side is that the more social strategy pays a higher cost on average of infection, while the sort of safe strategy doesn't pay that cost. So on the whole, they sort of balance out.
Before we talk more about the results, take us through how it actually works. How did you code this up? What does it do to model these scenarios?
It's a computer simulation where every individual is just a little bit of computer code. Each individual is essentially just made up of a set of preferences. What's your preference for being near other individuals that have food? What's your preference for being near other individuals that don't have food? And what's your preference for being near food itself? And this allows us to run lots of simulations sort of over and over again, and then compare what happens in every instance and see whether there's any general patterns to be found.
Do you pre-specify those choices that each individual in your group that you're studying makes, or are those fluid? Can they change?
Well, we do pre-specify them in the sense that they can't change over an individual's lifetime. But because we take this sort of evolutionary approach, all the possible choices that exist in the population, and we allow sort of every single combination that could exist to exist.
So
What
that's like your
happens
gene pool, effectively. You've got a range of possible
exactly?
behaviours in the population from which a selection may occur.
Yeah, absolutely. So what we do then in the sort of evolutionary simulation here is to say, well, we've given the population all the possible options they could have. So we have an initial phase of selection where that's whittled down to the preferences, or let's say the genes, if you will, that are most suitable for moving and socializing when there's no infectious disease. And then once we introduce the infectious disease in terms of sort of randomly infecting a certain number of individuals and letting them spread the disease to others, we look to see how that whittling changes essentially. So of the individuals that have made it thus far, which will continue to survive. And of course, it's a mutational process here as well, which means that new combinations can always arise. So there's never a permanent loss of any of the genes or any of the preferences or any of the combinations, as it were.
And so what emerges and over what timeline with the disease when you introduce some sort of contagion into the group? What solution does the group settle on and how quickly?
Yeah, so I would first start by saying that the group itself doesn't really settle on a single solution. I think that's what's really interesting here. Every individual is making slightly different choices given the same information, even if they all at some point prefer avoiding their neighbors, they will avoid them to different extents. The two main things that we want to really focus on here is whether they avoid all their neighbors or whether they're willing to associate with some of them given some benefit. And that benefit is the social information, as it were, the sort of information of who's been successful and where. In the main results of our paper, what we see is that the population essentially splits into two morphs or two types of individuals. Those that play it really safe and avoid everyone else, and then also get infected much less. And then these others, which are willing to take the risk of being infected, given a certain sort of benefit, which is again this useful social information, and then pay the cost of being, on average, infected more frequently.
So, bottom line then, what do you think this study adds and how does it change or influence our thinking?
Yeah, so I would say that because this is a theoretical biology study that's really based on simulation models, what it does is that it adds another sort of toolkit to understanding potential scenarios for the future, especially long-term scenarios that can help with sort of not just management, but also understanding what the potential outcomes of letting infectious diseases spread to novel host populations might be. Which is not to say, of course, this is exactly what will happen, but rather it helps to set the, let's say, to set the tone for discussions around that sort of scenario. So again, to sort of come back to the avian influenza pandemic, it's panzootic. It's one of the things that perhaps people should consider when thinking about how to manage that, which is that there could be very long-term effects on how social the survivors of this outbreak are likely to be, and what that has, what that means for sort of management of sort of populations of conservation concern. But again, I will emphasize that this is a theoretical study, which is really useful, I think, for the setting the parameters within which one can interpret data that one collects from the real world in the future, or reinterprets data that have already been collected.
From the London School of Hygiene and Tropical Medicine, Pratik Gupte there. In medicine, we often use a person's chronological age, how many candles are on their birthday cake, as a guide to disease risk. But what really counts is a person's biological age. How well lived in is their body? A person who eats a poor diet, smokes and breathes bad air is likely to age much faster than someone without those exposures. And we can read various biochemical clocks, such as chemical changes to DNA, that do correlate well with these sorts of insults, at least in adults. But what about in children? Are these markers helpful in that context, or does the process of development and growth make them much harder to interpret? Imperial College's Oliver Robinson has been trying to find out.
We had a very nice study, and actually I was quite involved in the early part of my career in getting it off the ground, but it was a pan-European cohort. So now these children have been followed. Currently, the data I looked at, they were between 5 and 12 years old, but we had information since the pregnancy of the mothers of over 1,000 children with all these different biological and developmental measures.
What were the measures?
There are many aspects to development. We looked at their height, we looked at fat mass, we did a range of cognitive tests, general fluid intelligence, and another test, which I really enjoyed watching the children do because it's extremely boring. They basically have to look at a whole screen of fishes and click if they see a fish pointing the other way. And you can see the children starting to squirm and wriggle because they're getting really bored. But that's the whole point, is to measure their attention. We looked at behavior, tantrums, bullying, are they kind to each other? And then finally, we looked at lung function as well.
What about biochemical markers like DNA methylation marks and so on? Were you appraising that too?
Yeah, so we had 4 different markers actually: DNA methylation, telomere length, and then we measured RNA, basically which genes are being read. And finally, we looked at a range of chemicals in the blood, big molecules, so we're talking about proteins and metabolites. We combined all of these circulating markers, the proteins and metabolites, and use this to develop a sort of average profile of children at different ages.
And when you do this, what trends emerge? Can you see a clear aging specific pattern? Do the markers hold true to what we think is the genuine age and developmental pathway or track these children are on? And does it line up with what we see and have learned about adult aging?
I would say in many ways it does actually. As we know from many, many studies, that telomere length and DNA methylation age reflect unhealthy aging. And in some respects, we do see that it is harmful for the development of children. When I say harmful, I mean that actually, if you look at their behaviors, so how often they have tantrums and so on, this was greater for the children with the advanced biological age from these measures. That is showing that it's not really very good for their development because they're actually quite immature, perversely, if they have a mature DNA methylation age. So I think actually what is really happening with these measures is an unhealthy environment is causing changes to these chemicals. I say this because we see for DNA methylation clock, I mean, if the children's parents are smokers, they have an older DNA methylation age. And similarly for telomere length, if the parents are less wealthy, which might reflect a less beneficial environment, they actually have an older telomere length. So for these markers, it seems that, as in adults, it's probably not very good for children to be older biologically. But for some of the other markers we looked at, the chemicals circulating in the blood, here actually it seemed to reflect developmental maturity. So we saw a strong association between older biological age using these markers with their working memory and their fluid intelligence.
So you've got a certain suite of markers that appear to be quite good at telling us about developmental progress. You've also got a set of, or a suite of markers that seem to be quite good at pointing to exposure to potentially deleterious environments. So actually you could integrate both of these, and you can ask, in the same way as we plot, say, head size and body weight on a growth chart, you can ask, is this child developing according to where we would expect them to develop, but also what insults are they having to weather as they go through, and are there any things we can therefore try to put right?
Exactly. And I think this is what's intriguing, is showing that if they're less affluent and maybe there's a lower education environment in the family, which is all not optimal for children's development, we find that actually it has a real biological impact. So it is showing that it's really important to make sure that all these children have the best start in life.
Oliver Robinson. And that is where we must leave it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. There are previous editions, the references and also full text transcripts for these programmes, as well as details on how you can subscribe to this podcast at nakedscientist.com/eLife. The Naked Scientists also publish a weekly science programme covering the latest leading science stories, and you can find that at nakedscientist.com/podcast. I shall be back in another month with another look inside eLife. But until then, from me, Chris Smith, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.